Hello, my name is Dr. Dewan S. Raja. Today I will discuss about the histology of the capillaries. Capillaries are the smallest blood vessel. They are composed of single layer of squamous epithelium. This is the squamous epithelium. They are called endothelial cells with no smooth muscles. The endothelial cells are surrounded by the basal lamina. We'll also get pericyte in certain places. This is the pericyte, and this pericyte is a mesenchymal cell. They have processes. Their number is variable, and they regulate the flow of blood through the capillaries. Also, they can repair the damaged capillary by means of conversion from the pericyte to the endothelial cell and to the connective tissue. So we have the basal lamina here outside these cells, and basal lamina is formed by the secretion from the endothelial cells. So this is the endothelial cell, this is the pericyte, this is the basal lamina. This is one RBC is passing through the capillary. The capillary diameter is about 7 to 9 micron in case of continuous capillary and fenestrated capillary. In the discontinuous capillary, it may be up to 30 to 50 micron. So this is the identifying point, like that endothelial cell, basal lamina, and we may get pericyte. Okay, so we have three types of capillaries: continuous capillary, fenestrated capillary, and the sinusoidal or discontinuous capillary. The capillary has pinocytic vesicle here, especially in the continuous capillary, and these pinocytic vesicle are essential to transmit substances from the capillary to the surrounding tissue by means of a carrier by pinocytosis. This is the endothelial cell, this is the RBC again, this is the basal lamina, and we may get a few pericytes. The endothelial cells are connected to each other by tight junction or junular occludens that regulate the flow of substances, that also regulate the flow of substances from inside the capillary to the surrounding tissue. This is the fenestrated capillary. We have a lot of fenestration, but these fenestrations are not homogeneously distributed. They are scattered, and they are covered by a diaphragm except in the kidney fenestrated capillary. This is the sinusoidal capillary. We are seeing gap in the continuity of the endothelial cell, and the basal lamina may be absent in many places. The continuous capillaries are the most common type of capillaries. They are found in the muscle, connective tissue, nervous tissue, and exocrine gland. And the junction between the endothelial cell is the tight junction. And if we have pericyte, the pericyte has their own basal lamina that fuse with this basal lamina, or also they may share the same basal lamina with that of the endothelial cell, and the pericyte has junction with that of the endothelial cell by means of the gap junction. So there may be gap junction between the pericyte and the endothelial cell. The junction between the endothelial cell is the tight junction or junular occludens. Okay. Fenestrated capillary, as we mentioned, we have a lot of fenestration, and that is covered by the diaphragm, except in kidney. Kidney glomeruli has the fenestrated capillary that is not covered by the diaphragm. The diaphragm is ultra thin structure that cover the fenestrated capillaries except the fenestration of the kidney glomeruli. Okay, this is sinusoidal capillary. This is white. It is tortuous. Basal lamina may be absent in many places. There is discontinuity of the cells, the endothelial cells. Okay, that is very common, and the flow of blood is very minimum. And we may also get macrophage both outside and inside the sinusoidal capillary. Okay, this is the capillary bed, a part of the microcirculation. What happened? This is the arteriole, this is the venule. So we have capillary bed that connects the arteriole to venule. We have the meta-arteriole. From the arteriole it goes there, and we have the pre-capillary sphincter. These are the true capillaries. These are the pre-capillary sphincter. So not necessarily all the capillary is always open. So it is regulated by means of this capillary. And also if it is closed here, then blood may go from the arteriole through the meta-arteriole directly to the venule through the thoroughfare channel. The thoroughfare channel has no precapillary sphincters. So these are the true capillaries. So here sphincter mechanism is working. So blood is going directly from arteriole to venule. So we'll have some highlights about the capillaries, types of capillaries. We have three types of capillaries: the continuous capillary, the fenestrated capillary, and the sinusoidal or discontinuous capillary. The fenestrated capillary in the kidney is little different. It has fenestration, but there is no diaphragm over the fenestration. Location of each type, yes. Continuous capillary is present in the muscular tissue, in the connective tissue, in the nervous tissue. That is very common. Fenestrated capillary is present in the intestine, in the pancreas, and in the endocrine gland, and also in the kidney glomeruli. We have fenestrated capillary, but fenestration has no diaphragm. Pericyte are mesenchymal connective tissue cell that covers the endothelial cell with multiple processes. The number is variable. They are not present everywhere. They can control the flow of blood through the capillary. Also, they can be converted into endothelial cell and connective tissue cell in case of the repair process of the capillaries. Tight junctions, the junction between the endothelial cell, that allow the variable entry and exit of the substances from the capillary to the surrounding tissue. Gap junction is present between the pericyte and the endothelial cell. Pinocytes are mostly seen in the continuous capillary. Pinocytes are the vesicle for the flow of water or fluid through the cell membrane by means of the carrier. Fenestration are the gap or small opening, maybe large opening in the sinusoidal capillary. Capillary bed is the collection of capillary that connects the arteriole to venule. It also includes the meta arteriole and the thoroughfare channel. So capillary bed in along with them together forms the microcirculation. So capillary is not always open. It is controlled by the precapillary sphincter present in the meta-arteriole. Meta-arteriole are the direct branch of the arterioles. Okay, these are my references. And that's all about the histology of the capillaries. If you have any question, please feel free to ask me. Please share the information with your friends. And please support my channel. Please subscribe me. And have a nice day. Bye now.